Hi, everyone. I'm not sure if we're still waiting on more people to uh, move from the previous session to here, but maybe I'll wait a little bit longer. I'm actually not sure um, how many there were, how many people there were today in the previous session. 24? Okay. Maybe we can just wait another few minutes to get everyone in here, and then I can start the session.
Ted. And do you think you need Ivan for this session? I can call him and let him know if you want.
I don't think we'll need a bond for this. Yeah, I think yeah, Bob won't be here for this session, but I'm going to lead us through the module 5, and that's working for today's advanced topic. So yeah, I guess we'll just start. Today's advanced topic will just be working with our own images. I know Bob gave an extensive talk about all the functionalities of CellOrganizer and a lot of the tests that we have been doing. We've been showcasing image files that we have, or like the founder files for CellOrganizer. But I'm sure for a lot of people that are participating, they're more interested in using their own images and seeing what they can get out from CellOrganizer. So that's what we're going to be doing today. So let me share my screen. And then everyone should have a link to this module 5. It's a Google doc right now. So the beginning part is going to be a little bit more of just how like image segmentation works in general and what we're going to be using. This is all, if you want to think about it, kind of like pre-processing your data. As you can see, we're going to need these three kind of software, Fiji or ImageJ. This is just to visualize your images and to kind of like crop them and shape them so that they can be properly fed into a segmenter. And the one that I chose to use is CellPose. I know there's a lot of different autonomous cell segmentation softwares out there, and feel free to use whichever you want as long as you can output a segmentation mask for your image that is in a TIFF or OME TIFF format, because that will be the required format that CellOrganizer takes. And again, this link is in I believe is in the CellOrganizer tutorials. So you can check this out. It has very detailed instructions about how to install this software. It gives you the links to these softwares, and they will have like the instructions there. So
yeah, please could you post the link to this document to chat?
Yeah, of course.
Not so straightforward to find. Thank you.
It's a great idea. Let's see. Where's chat? There it is. Okay. So this is the link to the documentation chat. And so we'll properly start now. So for So let's start with using ImageJ to pre-process getting the files into the correct format. So let me start up ImageJ. For those of you that aren't familiar with ImageJ or Fiji, it's just file format reader. It uses file formats to read in like images, PNGs, any kind of images you think of, CZI, etc. So it's got a very extensive reader for all kind of image formats. Give me a second. Let me try to open up Fiji. There we go. There it is. So let me share. Actually, is there a way to just share my desktop? Oh, there is. Okay, great. So it's perfect because I'll do that. So then I don't have to change between views. So this is ImageJ right here. It's just like, it's the GUI is not super intensive, but let's open up a file that we're interested in. So I'll walk through, I mean, in the module, there's a bunch of instructions. So, you know, because everyone's image files might be different. Some of you guys might have multi-channel, you know, 3D images. Others of you might have like 2D images that aren't multi-channel, that are singular channel. Ideally, we want to get all, and you know, you might have more than one file. Ideally, we would want any kind of image files that you want to process, get it into all one format. That's kind of what we have prepared in like section A is like a single file for each view containing a 4D image of one or more fluorescent channels. So this is ideally what we will want to get your image format to be. And then proceeding, like if you have multiple files containing all channels or Z slices for each Z slice or channel, then or you have like somewhat of a 3D image, then you have to go through some of these pre-processing steps to get it into the A format that we would like. So, you know, depending on what kind of file you have, you'll follow the instructions there. But for today, we'll walk through maybe just a simple example of something that you might see. Say we have one file for each channel or so. So let's open up image file. There we go. Image for segmentation. Let's open up. Do we want this? I think we would not rather have that. Let me try to that. So I was creating this beforehand. So we don't want that in there because that might mess up what we're trying to do. So trash. Okay. So back to downloads, segmentation. Let's just start from here. Okay, so we want, okay, great. So let's open up, so you want to go to, if you have this kind of like one single file for each channel, then it's kind of like you want to sequence the images. So you want to go to imports, image sequence, click on the necessary folder in here, and then just click on the first one. And then this option pops up. It says how many number of images. We had 4 in there. What do we want to start with? These kind of don't matter, but increments does matter because what happens here is it sorts that directory, and then you know, you can start from the index of 1, 2, so on. But usually you just want to keep this consistent. Put all the images that you want to like concatenate into one folder, and usually you don't have to set any of these. Just press OK, and then you'll see your images here in this window viewer. So as you can see, I have cells. This seems to be the cytoplasm, cell membranes, or fibroblasts. That might be it. My biology knowledge is not all there. But yeah, so you have 3 different channels. I believe this is the nucleus. And then, so now that we have these 4 channels, let's look at some of the properties. Make sure this is what we want, right? So it says here that channel is 1, Z slices 4. Time frames is usually like just like if you have like a time series data. Usually this is always 1 for the images that we process. But so as you can see now, although it's very extensive in the kind of ImageJ and bio formats in the reader, it sometimes will read the metadata or like your dimensionality is wrong. So what we can do here is we can reorder it. So we would go to image hyperstacks, let's go to reorder hyperstack, right? So we know that we had 4 channels we wanted, right? Those were separate files. Those are 4 separate files for each channel. So, and we know that this is a 2D image. And don't worry, we'll get to 3D images as well. I could load up a sample of that, but I just wanted to walk you through how to troubleshoot or troubleshoot some of the images that you have. So here we would just say like number of slices. So we know that we want the channels to become Z slices, and we want the slices for Z to be channels. The time frames is the same. I want to say okay. Now we can see a better picture of this, right? Now as you can see here, this is also kind of ordered wrong as well. So we can do another troubleshoot here, in which we can. I mean, this is more for more just for aesthetics, of the actual image. But what you can do is go to images. Let's see. I think it's colors.
I'm sorry. Can we download the images somewhere?
Excuse me.
If we can get the images somewhere that you have.
Oh, okay. This was just kind of a sample that we were doing. I can also, yeah, the images unfortunately is not available, but
Okay. So I thought that we should follow or do the same thing on our side. Sorry.
I see.
Okay. It's okay then.
I guess the idea was that they would have their own images that they would use, and this was just kind of a walkthrough of like kind of the steps if you ever encounter it. But it's more so like if they wanna just do the modules themselves, they're feel free to do that because they have the instructions of pretty much what I'm doing right now. But Adam, do you want me to give you the images that I'm using right now, or I guess it's kind of too late?
Yeah, like I'm okay. I just thought that I could follow with you, but yeah, it's okay. Don't worry.
Okay. Yeah. Sorry about that. For anyone that was trying to follow along through this, I wanted to use these images as well. This is more just an example. But I would try to think the images that we do provide in CellOrganizer when you build a container, any of those are also usable for this. To be honest, I don't think I should probably have used some images that are in there, but this was just images that I had very readily on hand. That was that had like a nice clear picture. So so apologies for not providing these for anyone who wanted to follow along.
Thank you.
But yeah, let's.
So sorry for interrupting.
No, that's fine.
Sorry, Ted, could I ask?
Yes.
So what were the accepted model file formats? You mentioned them quickly, but I missed that. So I have model file, but it's at this kind of MATLAB format .model or whatever. I don't know if that will help me.
Oh, so that's a
segmentation.
So for the model, so that's after you process through CellOrganizer with your images. Right now, what we're doing is just taking.
Yeah, no, sorry, I meant I have a segmented image, and the what I'm talking about is the segmentation model. It's a .model, whatever MATLAB file format that we have, and I was just thinking if I can use it or what the format must be.
So for the segmented mask, the format would have to be. I mean, you can have it as an OME TIFF. That's the preferred format.
Okay.
Yeah.
Around then.
Yeah, but we so if you guys are going ahead, yeah, feel free to go ahead. I'm just kind of walking through this step by step. But yeah, if like segmentation mask, you want an OME TIFF. There is actually, but yeah, I'll get to it later. I'll try to breeze through this. But yeah, so once you have your images like this, just like if you want to just maybe for aesthetic reasons, you want to make sure that you have, the red channel and the red, blue channel and blue. Then what you can do is, I believe it's arrange channels. So you see this arrange channels. You see the first one, it says it should be blue, so we can put it, or we can put it into, a blue coloring. Second one, I believe this one is cytoplasm, so it should be green. And then this one is wait, no, sorry. This one is red. Yes, there we go. And then so we have it in these new channel orders. Say OK, and then this spits back us a new image. So now, just for sanity's sake, let's just check the properties again. And now we see that the channels are 4, C slice is 1, frames are 1. Okay, so we're good. So then we just go to save as a TIFF, and then we can save it in the same folder. It doesn't really matter. Just make sure you know where it is. And then now we can start segmenting this image, because right now it's just image that we got from the microscope, and then and then now we want to use it through a segmenter. So I mean, there's a bunch of different segmentation methods like that. I feel like it's an open-ended field right now of how to segment image, like computer vision in general. But the one that I chose is a pretty popular one currently. CellPose, it does have its bugs and issues, but it's relatively intuitive to use and pretty simple for new users. So let's go to, okay, so another thing is, I'll just show you the GUI, but let me go to, let's just open up CellPose right here. So I'll show you guys the GitHub repo of it. And this is CellPose by Carson Stringer. Her team wrote the segmenter, and in here, like in the README, you can have all the instructions on how to install it yourself. If you don't, they also provide, using Google Labs, kind of, if you don't want to, like, this is kind of like a Jupyter Notebooks. If you don't want to go through all those installation yourself on your local computer, say you have a really old computer, then a lot of this might be hard to run on it. For example, I know, sorry, let me share that again. For example, I know that they have pretty hard requirements for your PC to be able to run it. Don't remember where that is, but I believe you have to have like 8 gigabytes of RAM. So long story short, it's like if you have an older computer, it might be better if you use Google Colabs, because in there they also use a GPU setting, which actually speeds up a lot of the processing. But for now, we're gonna just look at the GUI version of this, because it's a lot more intuitive. They also provide source code in which you can just directly, using a Jupyter notebook or something, type in, they have an API for this. You can just type in code for it. If you don't want to, if you're pretty well versed in programming, you don't want to use like the GUI version. But for simplicity's sake, this is the GUI version. So what we want to do is now we want to load in that segmentation. So let's see. Where did I put it? Okay, here. No. GFP. So, okay, so now you can see it loaded up in their GUI. It's pretty much the same representation that we saw in ImageJ or Fiji. And right now you can play around the setting. I'm not actually going to run this because this definitely takes a very long time to run in general. But we can walk through some of the settings so that you guys could play around with it. Or I'll also go to their Google Colabs page and showcase some kind of segmentation that they use and walk through the code that the language that walks through like the API they provide as well. So, but if we're going to use the GUI, pretty much like the views that we have is RGB. It's just red, green, blue channels. You can see it here. Right now the blue is the membrane, red is the nucleus, and then green, I guess, is like something like maybe some stuff in the cytoplasm. I'm not quite sure. I would think it would just be some stuff in the cytoplasm, I guess. But right now, the two things that you really need to know for using this segmentator is just that it needs to know what the cell diameters are. What you can do is input this manually. They also have a way to calibrate itself, in which it looks for the average, I guess, cell diameter in pixels here. So I mean, you can click, calibrate, and you see here it's using CPU to do it. So this is going to take forever. But I mean, when you load this in, it has information too. It has the image is going to be a 2048 by 2048 image. That's 3 channels, and it has 1 Z slice because it is a 2D image. So this is the thing that unfortunately this is going to take a pretty long time to do. But after you calibrate, it usually will adjust it so that you have what they think what the cell diameter is for your image. Now, that might not be accurate, and if you have a better number, maybe you're more familiar with your image data set and you know what the average cell diameter is, you can input that manually and it doesn't have to try to calibrate by itself. So once you do that, you can go down to the model. So the model is like, what are you trying to segment? So they have an option for cytoplasm. So unfortunately, this is running right now. So we have to wait until it's over. Or maybe I can just kill it. Oh, no, it's done. So it says, so for them, they think this is 27.8 pixels, and that would be your average cell diameter. Great. So let's go down to models. So you can either segment the cytoplasm, nuclei, or cyto2. I'm not really sure what this is. This might be a new functionality that they have. Maybe like a newer, this is a new cyto model for user submissions. So a cool thing about CellPose is that they take user submissions and put it into their cloud servers, in which they can use to reevaluate models and update it as well. So for us, it's like, okay, so we want to use maybe we'll just use their old cyto model, and then this is the channel to segment. So okay, well, so what would you be interested in? Would you be interested in segmenting the nucleus, the cell membrane, or something else? But we would have to specify that channel. Now, for us, I believe the blue channel would be maybe the cell membrane, but maybe let's just segment the nucleus. Now, the one thing that I know that would probably give this segmenter issue is these holes, if you see it here. Like you have the nucleus membrane, but it's not completely filled. So this might give it issues because depending on the algorithm that it uses, it's not just, it's very specific. So any holes or any discoloration, it would have a hard time finding this envelope. For us, it'd be pretty easy to segment, right? We would just manually segment this part, but it might be harder for it to segment. So a lot of your images, you don't want to have, I guess, these holes right here. But regardless, you set this up and you click run segmentation, and then it's gonna say finding mask. It's gonna take a while to run. That's the thing. So let's just move on to their notebook then to give you a better image of things that have been run. Cancel this out, and then let's go to the notebook. So this is for 3D images running on CellPose. So as you can see, what they have here is like an original image, and I guess these are some of the predicted outlines they have. They have like a few examples of all of this, and then what we want to do. Pretty much this is going to be the predicted outlines right here is going to be what the segmentation would look like after it's run. And, depending on however many cells, how big, what the resolution is, that if you're using CPU, GPU, it could take a while for this to happen. But I mean, obviously this looks super great because it's one of their images that they've been using. And you have other options to display your predicted masks. But one of my recommendation is if you don't have a great computer, you can use this Google Colabs notebook. And I will also provide this link as well. But let's go to the code that I actually wanted to show you guys. It's just this run CellPose 3D. So once you have installed CellPose, you can actually just use it without the GUI interface. And pretty much all you would have to do is you have to load in your image file. And for here, it's this image 3D file. And we're saying, what they're saying is they want to use everything, you know, they don't want to cut it anywhere a certain way. So this image 3D file is going to be in the structure of an array, a 3D array for 3 dimensions. So pretty much you just want to say model.evaluation and the parameters, then you want obviously want to put in your image file. And then for channels, these are the channels that you want to segment, you know. So for us, we had like what, 4 channels. Say we only wanted a nuclear membrane and cell membrane, right? We would choose, we'd specify the channel. Now this is indexed, so we would know that going back to maybe this picture, right? We know that, so for the blue, right? Depending on how this is ordered, the blue is going to be the cell membrane, nuclear membrane. So we might, for here, we might say 1 and 2, right? And then diameter, this is like what we were talking about before when they were going to calibrate. If you look through the API, I'm sure you can also not set diameter and it'll calibrate it itself. But if you want to specify the diameter, so say that you're familiar with these images, then you can also do that. And then there's also do 3D equals true or false. So for the images that we used before, we would say that's false because, you know, we don't have 3D images, we have 2D images. And then this would be something that would be similar to what you would have seen in the terminal, and it would just be processing, it would just be running this. Finally, it would give you the segmented mask, and then you can see it right here. So feel free to play around with this. The good thing about Google Colabs is that you don't really need to install anything onto your local machine. Everything's already pre-installed in Google Server. So you can just start kind of running this from scratch. So I could say, new code line, I could do something, and I can, you know, I have to make sure that it's the same thing as like, it's the same thing as the Jupyter notebooks. You know, I would have to run these. I would have to run this, right? Make sure this is finished, run all the imports, and then I can start coding in here myself. So it's a great feature for anyone who is trying to do this with a computer.
So Ted, would it be possible to segment just a single instance of a cell and make a cell shape from those single images? Not a learned shape, but just a reconstructed shape, and have that and output that as an SBML spatial file.
Yeah, I think you could probably do that. Yeah.
Because of course you need the 3D, the confocal slices in the optical sections in 3D, of course.
Right. Yeah, I believe you could do that. I'm not.
Yeah, because what if certainly a generative model is, you know, is fantastic for all the reasons, right, that you said before. But what if you wanted to have a model of a specific instance that you see in your data set, and you have the image of it, right? And you say, I have the shape space, but I want to model that one right there. And so if you could do that, then you wouldn't necessarily have to even, you know, learn a model, because you have the instance in front of you.
Right. Yeah. I mean, intuitively, I think that's possible because you're just saying like, do not learn a generative model. I have what I needed in this actual image itself. Let's bypass all of that.
Right.
Now,
Right.
if there's a parameter that does that, I'm not super certain. That would be a great question for Bob, but
Okay.
in my mind, that it would, it would be skipping a lot of steps in CellOrganizer, just outputting the
Sure.
data because from
Sure.
directly from the image. But yeah, I mean, that's a great question, Tom, that I can
Okay.
ask Bob about.
Okay. Sure.
But I would think it's possible, but I am also not, the person that have spent 10 years or 15 years running all of CellOrganizer, but I'll write that down as
Sure.
a question. Yeah.
Sure. Okay. There could be lots of good reasons for wanting to do that, I think.
Yeah, and that goes back to exactly what Bob was talking about, that these are like, and if it's not a feature, we should probably make it a feature in CellOrganizer. So
Yeah.
that's what I'm saying. Like, if there's features that you guys are interested in having, definitely use that contact us page and describe it, and us, the programmers, will try to make these dreams into realities for
Yeah.
CellOrganizer. But yeah, great question, Tom. Okay, so I guess moving on. So
Okay.
once we have the segmented image, okay, so in my notebook, I do give you kind of an example of a 3D image that it was segmented. You can see here in this picture. Now, this is using an older version of CellPose. The GUI kind of has changed, but it's the same. It's the same premise in the sense that you kind of, I think I see that you kind of just load up your image once you have pre-processed it in here, and then just calibrate it and then run the segmentation. Now, once it's finished, you kind of see these overlapping, I want to say like different coloring, right? You over your cells, and this is what the segmented mask. This is from this is what their model has predicted the cell shape to be. And in here, I think my model was Cyto, and so since these weren't RGB, I guess these were just a 3D image with one channel that we would be using the gray channel to segment here. So you'll see something like this, and then what you want to do is maybe we can open it back up, I guess. But I mean, it's pretty much just once you have the segmented image, you want to save it as a TIFF. And you just go to File and Save, just like any other application has that ability to. So now we finally get into the last step of generating OMI TIFFs with regions of interest. So this is very important because this is what CellOrganizer prefers. I guess CellOrganizer prefers the file type to be. Now, I know in module 1 we gave you a bunch of different ways, but at the very end, I think OMI TIFFs with regions of ROIs is our best supported file type, and it's readily easily to read. It makes your life easier because you kind of only have to just upload one file into CellOrganizer instead of the different ways that we showed before. I'll go back to module 1. Let's go back to module 1. That's demo, sorry. Where am I going? There we go. In which we say, oh, hey, there's single file for each cell and channel, multiple cells and single channels with binary mask. And this is kind of like really inefficient because pretty much you're uploading the same images again and again, but the masks kind of change. So since the binary mask, say you have 3 cells in your images, it would be pretty much the same images over and over again, but in your options mask, it would be the different cell masks. So I mean, it's not as efficient as if we were just going to go with OMI TIFFs files with regions of interest. And for that, all you pretty much need is just the cells, right? Like, or just that one file without a mask, because the mask will be in the metadata, which is called regions of interest. So let's go back to this notebook. Let's reload this. Maybe. Okay. There we go. All right. So now we're getting, so this is the step right after we have, used our segmenter, processed mask for it, and we have both the corresponding image and we have the mask file. So what we're going to do from here is so for here, I've just written some code. These are just functions, kind of like imports themselves. So you're just going to run this notebook, and this notebook is going to be in your helper notebooks. So you just run this, acquire all the functions, and then for your input directories, you want to run this line and then say, okay, so pretty much this is asking you for like, where is your image at? And provide me the directory for it. So since I didn't prepare ahead, I kind of have to find where the image directory is. Now, this might or might not work depending on, since this is outside of the container, let me actually move it inside the container. That would be a good idea. Since everything is in the scope of the container. Test images. Let me just copy. So let me copy these two directories back into my BIOS. Okay, I'm gonna show you guys it. Go back to BIOS and maybe just put your segmented image, the image that you're using, and then the segmented image in the BIOS container. And then this says 2 items. We go on this, I think. And then test files. I don't know why this is here. Give me 1 moment. Test max. This is the one we need. Okay, so as you can see here, we have both our test images and test masks. That's just the folder that I called it. And then we want to say something like, let's see, maybe we want to, give me a second. You can also open a terminal here. I just want to know where the root is. So CD root should be CellOrganizer, right? Yep. So then you want to do work. That's weird. I don't know why. Should be in local. But it doesn't have anything here. That's strange. Oh, there we go. Okay. So we want to copy this path right here, pretty much. And we want to go back to here and put it into as a string. So local, we know that once we get into local, what we have, yeah, test images and test masks. That's what we're looking for. So we want to say, since this is the image directory, I have my image, put the image directory in here. And then once you have done that, press enter. Now, pretty much what this does is that the image directory path is going to be pointing to this string that you created here. do that same thing for the mask file. And so if you're using the container, this is exactly what you're going to be doing as well. Instead of images, put it as the name of your directory. Go. And then run the cell below to check the pathing's correct. So this is just a sanity check, even though I kind of already, this is what you, well, this is wrong actually. Let me do it again. Because there was one too many quotes. And then there's a sanity check, you run it to print it. So just to make sure that the pathing's right. Yep. And then let's write out this OMI TIFF. So we just run this last line. And ideally, this would be in our results. It should have given you some kind of output path, I guess. Here. Image OMXL. So this is finished running. Let's check our notebook. There's our notebook, helper notebook. But yeah, so this essentially would have just written out your OME XML all together in regions of ROI. So yeah, let me find it for you guys here, and then I'll show you what it looks like. So here it is in your image directory. So yeah, so in your images directory, it will have process. So I've done like done this 3 other times, but it would have written out your OMI TIFFs with ROIs. And what we can then do from here is just go back to module 1 and uncomment this. Put in, so I already have the directory, so put it in, your directory wherever the OMI TIFF was created, which should be your images labeled under OMI TIFFs with ROIs. Now I'm going to delete, because I had 3 in there, I'm going to delete the 2 older ones and just use the newest one that we created. Or I could do, I guess what I could do is I guess let me just delete this. Will be simpler. And then we can use that and run it to image to SML. So finally, we're getting back. Let me just run the rest of this. So again, before, you can specify whatever parameters, what kind of model file that you want to generate, like what kind of generative model you want. Well, these are basic parameters like the file names, but here you can specify it. And for right now, I'm just going to use the basic settings we had before, because I don't want it to run for that long. And run this, kind of like we did before. And then you can see it working. And this is from, so yeah, it ran pretty quickly because we were doing a pretty basic ratio model. And then that's pretty much end to end of how you get from just your image, getting into OMI TIFF structure, like segmenting it, getting into OMI TIFF structure, and then finally importing it back into CellOrganizer to make a model out of it. So is there any questions that you guys have?
I have one question. Do you mind showing the images as an output?
The images as an output?
Yeah, whatever images you get. I'm not able to do it from my side. Could you show the images?
So which image do you want me to show?
Like whatever output you got.
Oh, okay, like the output of out of image.slml?
Yeah.
Okay. Yeah, sure thing. So what you would do is just go back to local and then go to results. You should have something in module one, because that's where we ran it. And this would be your module one.mat file. Here's a log of pretty much everything that was on your command line. But it wouldn't be an image, it would just be you know, the actual model file, model.mat file with all of the instructions, or not instructions, but with all the descriptors inside of there. When we ran through the other module, we learned ways of how to display that, right? There was one way to read it, I believe Bob showed you guys yesterday. We had another notebook. Sorry, let me mute this. We had another notebook that kind of displayed all the descriptors for this module, and we were able to generate that shape plot. But yeah, that would be the output of this.
Right. Right.
Yeah, so you can kind of monitor whatever that notebook was that allowed us to print out all of it. Failing to find it right now, but I'm sure it's in one of the. I'm sure it's one in one of these directories. Yeah.
Okay. Got it. Got it.
Is there any other questions? I guess, does anybody have images of their own that they're interested in using? And like, if you want to share like what kind of like type of image it is, the dimensionality of it.
I do have my own images. I don't know if it will be, it might be a little cumbersome to do this. It's a stack of images. I don't know, and I kind of lost the track of using CellPose, and I never do this OME TIFF files. So getting the segmentation model might be now a bit cumbersome. Maybe we could try it later.
Yeah, feel free to reach out to me. Yeah, I think that part is probably most confusing because once you have your own images, using it in Fiji, getting it stacked together is relatively easy. But
I don't use Fiji in this case at all.
I see.
But maybe I'll try it later.
Yeah, Fiji's pretty intuitive to use.
Yeah. I use it a lot, but for the segmentation, I don't use it at all, and I don't produce. I can make TIFF files maybe. Are those okay?
Yeah, that's fine. Well,
fine. I don't know if they will work. But it's MATLAB based segmentation, and it produces this dot model, and I tried to convert it to TIFF. It did something. I don't know if it's correct. But that's how far I got with that.
Okay, but so wait, you said through MATLAB the image was already segmented, or you just? Sorry, I lost you.
Yeah, yes, the what do you call it? Mask, the segment, the model mask. It's dot model that is MATLAB can read, but I tried to convert it, but I don't know if I was successful, and
Well, if you already
So that
have
was my
a
issue.
segmented, okay, I see. So you already have a segmented mask.
Yes.
You also
Yes.
have the corresponding image as well.
Yes.
And it's in a .mat file. That's
Yes. Yeah. 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 I'm
okay.
not sure about the
So
format exactly. but-
that's interesting because ideally for your mask, you want it to be the output of cell pose is going to be an indexed image. So in the sense that it gives you the same resolution. So for in your case, you would have to convert that .mat file into an indexed. It should be probably what I'm thinking that mat file is just going to be an array of indexed, or it's a bit mask or an index mask in which you know, it goes from 1 to however many cells there are, and it's going to be the same resolution as your native image that you used. But instead,
Yes.
where there's going to be intensities, there's going to be 1 to however many, you know, like cells there are in that image. So all ones would dictate this is where cell 1 is, you know, in the resolution, and then 2 cell where all cell 2 is. So what you would have to do there is, I think there are in CellOrganizer itself, there is a way for you to write out, I think, a mask. So yeah, there's a lot of functionality in CellOrganizer. We kind of only touched up on the surface, but in the
Okay.
documentations,
Are those kind of things documented?
yes, I think it's in the documentation. So
Okay.
go to the website. Let me show here. Go to the website, go to documentations, and you're using the MATLAB version, or you're using, you said you were using MATLAB, right?
Not for CellOrganizer, no. Well, yeah.
Okay.
No,
Well, this is
for Docker.
okay. Well, this is kind of the documentations for MATLAB, and then there's an OMI TIFF one. Maybe it's not properly documented. Okay. I'll do a little digging for you, and then yeah, you can just reach out to me on Slack or something, and then we can
Okay.
probably.
Are you yourself familiar with the MATLAB, like how it structures or reading these? I have the models. I opened it in the MATLAB workspace and looking at it how it's structured. So maybe that would help.
Yeah, I mean, yeah, that probably would help. Just say yeah, send me a screenshot or something of
Yeah, I can do it in Slack right now if you
Okay.
want to look at it some point.
and then we can try to troubleshoot that, because then yeah, pretty much what you're trying to do is you're gonna try to extract that array out, an index mask, and then pretty much run that same notebook in the helper, this image format converter. Just run this notebook, specify the path of that directory, and then everything should be good. You should output 1 image. But yeah, I hope that helps. Does anybody else have any other questions? Okay, there's some stuff in chat. Sorry, I didn't see. Oh, no, that's still cool. Okay, so in that case, I actually don't know how long this session is. So for advanced topics, it's until,
I think it's scheduled to go until noon.
yeah, or
If you-
yeah, until noon. So
Yeah,
I guess my, does anybody, is there any other questions not pertaining to, you know, this module that anyone has questions about, or I don't know, like other issues that, you know, kind of like who that they have when trying to use their own images? I can just stay around, or if everyone wants to take a short break before the next section, take a longer break, I'm fine with that too. Just whatever you guys want to do.
I think I'm probably going to take a break until the next session.
Okay, all right, that's understandable. It's been a long 5 days. Previous break was a tad short. Yeah, okay. Well, I mean, in that case, I guess no one else has any questions. We can end it early here, take a longer break, and then I'll meet back up with everyone. I guess it would be 12:30, right? So
Sure, it's okay with me. So do you want me to close this session and like in 1 hour? When should I open the next one? Do you want to change the schedule, or?
I think we should stick with the schedule because that's what
Stick
everyone
with
is
the schedule. Sure.
expecting. Yeah.
So let me double check. So it's going to be 12:30.
Yes, that sounds right.
Oh, so I probably am not available to open it right then, but before I leave, I will open it and then make Caleb the host, and then he can make you co-host to be able to share the screen.
Okay.
Okay, thank you. See you then.
Okay. Thank you.
Bye-bye.
yeah.
Bye.
And yeah, I'm available over Slack for everyone who's typing in chat. Yeah, if you have your own OMI tips that you want help importing, I'm happy to do so. So I'll be around for any kind of those questions over Slack. Yep, but thanks, Rosita. Thanks, guys.
Thank you, Ted. That was great.
Thanks, Tom.
Yeah.